Okay. Good afternoon, friends. We have assembled here as part of the seminar series of Carlton and also the IEEE um, Engineering in Medicine and Biology Ottawa chapter and the CU at EMBS Carlton chapter as well, the student club as well, to listen to the talk given by Dr. Pino, who is from Neurovine. He's going to talk on the evolution of signal processing in EEG analysis. So let me do a brief introduction of Dr. Pino. Dr. Pino has a PhD from University of Waterloo, and he has been in this in medical device industry for the past 15 years in various roles, including product development. And he has 11 patents to his credit, and that fairly talks about his excellence in this area. Currently, he leads data science team at Neurovine, which is a local startup in Ottawa, and he leads a team of data scientists, and they are developing algorithms to help concussion patients to recover. Dr. Pino is very interested, has been working on very interesting projects. He has been involved in the heart rate variability study from smartwatches and then developing algorithms for wearable devices like Muse. So today we will have the opportunity to get his views on how DSP has evolved in EEG signal analysis. Now I'll turn on the podium to Dr. Pino. And Dr. Pino, it's all yours now.
Okay, great. Thank you, Shriram. I'm glad to be here, and thank you for that very nice introduction. So I'm just going to share my screen and get my PowerPoint set up. Okay, and so can you all see the slide?
Yes.
Okay, great. Is this in the way here, or do I have to move this out of the way? Okay.
Are you asking me? No. Okay.
Okay, great. All right, so I'm going to get started. Thank you for having me. So in this talk, I'm going to, sorry, I need to adjust one more thing. Okay, so in this talk, I'm going to review a number of digital signal processing methods and give a machine learning example, which is the current state of the art in classifying EEG signals. I'm going to talk about the evolution of analyzing EEG signals that span time domain, frequency domain, and frequency time domain analysis. I'll also provide an example of the current state of the art by using a deep learning method with convolutional neural networks to classify sleep stages. Electroencephalogram, EEG, monitors the electrical activity caused by the firing of neurons in the brain. EEG is relatively inexpensive relative to bigger machines such as fMRI, and it is non-invasive. One of the big advantages of EEG is that we're able to see brain activity as it happens in real time down to millisecond resolution. The photos here show two EEG systems. The one on the left is a full cap EEG system that can support dozens of electrodes. Full cap systems can go as high as having 256 EEG electrodes surrounding the scalp. The one on the right is the headband that I'm helping to develop at Neurovine. It will be used to help concussion patients recover more quickly. I'll speak about Neurovine some more in the last 5 minutes of my talk today. So I'm seeing some things here. Oh, is there chat coming through? Okay.
Don't worry about it. It's about the attendance.
Okay, great. Okay, so here are some examples of EEG signals. I'm going to provide some background on how the brain generates these signals before I get into the DSP part of my talk. So this slide shows how a single action potential happens along a neuron. So this is a diagram of a neuron. On the left are the dendrites that connect with synapses from other neurons. This large part here is the cell body and the nucleus of the neuron. And this long branch here is called the axon. It's the part that carries the action potential. On the right, that look like twigs, are the axon terminals. These synapse onto the dendrites of other neurons. When the neuron is at rest, there's a difference in sodium concentration across the axon membrane. So we can see here that there's a lot more sodium on the outside of the axon than there is on the inside. So the concentration of sodium atoms outside the axon creates a voltage difference called the resting membrane potential. And here we see it at minus 90 millivolts. So a neuron receives neurotransmitters on its dendrites that were released from other firing neurons connected to this neuron. When a dendrite receptor receives a neurotransmitter, it opens up a channel and it allows sodium to enter inside the body of the neuron. So we can see here the sodium entering inside the body of the neuron. When the amount of sodium that has entered the neuron reaches a voltage threshold, a cascade of voltage-gated channels opens in sequence, and the action potential travels down the length of the neuron called the axon. So you can see here that as the voltage potential changes here, it triggers the opening of another channel here. That other channel lets in more sodium, and on and on and on down the length of the axon. And if we were able to measure this, we'd see this action potential as voltage measured in the fluid outside of this axon. So at some point, the sodium channels close, and then sodium pumps remove the excess sodium, and the process starts over again. So yeah, the sodium pumps are like these little tiny molecular machines that are constantly pumping out sodium from inside the axon and removing it and getting it ready for the next action potential. So we can see here that the action potential lasts from, it's a spike, and it lasts anywhere between 1 to 4 milliseconds. So in this talk, I want to discuss the importance of neural oscillators. I'm going to use the metaphor of a water park bucket to illustrate how neurons can create oscillations. So the tap at the top of the bucket fills the bucket with water. And when the weight of the water is sufficient enough, the bucket tips over, unloading all of its water, and then a counterweight pulls it back to be filled again. So this is a metaphor for an action potential. The rate of flow of water into the bucket is proportional to the amount of sodium entering the neuron through channels that are open via receptors. So when the bucket reaches its tipping point, it will dump all of its water, just like when the neuron reaches its threshold, the neuron fires and once, and it's all or nothing. So just like the bucket tipping over, once the neuron has reached its threshold, it fires an action potential. So assuming the amount of sodium entering the neuron is at a constant rate, then the neuron will fire at a steady rate. The firing rate of the neuron is changed by changing the speed at which and the amount of sodium that enters. And so this is the most basic oscillator that can be found in the brain. So the heart is also an example of an oscillator in your body. And there are many similarities of muscle fiber to neurons as they both fire action potentials. In similar ways, the rate of your heartbeat is controlled by the amount of sodium entering into muscle fibers. So this simulation shows how a network of interconnected neurons can create a neural oscillator. Each blue dot in this top graph represents a single action potential that's fired. They're also called neuronal spikes. The summation of all these action potentials firing together here and here, etc., and then going quiet together here and here, create the peaks and valleys in this local voltage field here. And if there's enough neurons firing together simultaneously, then this voltage field can be measured by EEG, by electrodes on the scalp. So there are 3 different levels of neural oscillators, starting at the microscopic scale. So an individual neuron can create multiple action potentials called a spike train. And the spike trains can form many patterns. And the field potential created by a single neuron, it's too small to be picked up at the scalp by EEG sensors. So at the medium scale, which is sort of what's represented here, a group of neurons that are synchronized can create rhythmic changes. An interconnected network is comprised of excitatory coupling and inhibitory coupling synapses. So inhibitory receptors inhibit the firing rate of a neuron, whereas excitatory receptors increase the firing rate of a neuron by allowing in more sodium. So a large group of thousands of interconnected neurons, action potentials can be picked up at the scalp. And it's the balance and the combination of excitatory and inhibitory plus delay and feedback that can create oscillations on a larger scale. So the final level is the macroscopic scale. So neural oscillations can happen across different areas of the brain. And so when there's time delays coupled with feedback, then large regions of the brain can show oscillatory behavior. So in neuroscience, a major area of research is determining how oscillations are generated and what their functions are. So neural oscillations have been linked to information transfer across the brain, perception, motor control, memory, sleep, and awakeness. Note that the strength of the voltage field is a summation of the individual neurons firing. If the pattern of firing neurons is more scattered, then the amplitude of the summed action potentials become less stronger, and these peaks and valleys will diminish in strength. So here are some examples of EEG brainwaves measured at the scalp related to sleep stages. So at the top trace here are alpha waves, and they range in frequency from 8 hertz to 12 hertz, which indicate relaxed wakefulness. So this trace here is showing stage 1, sleep. And so stage 1 sleep is drowsiness. And it's often dominated by theta waves, which oscillate from 4 hertz to 7 hertz. In stage 2 sleep, which is light sleep, sleep spindles are found. And they're similar in frequency to alpha waves, but they're stronger and they appear all over the brain simultaneously. So stages 3 and 4 is deep sleep, which is dominated by the slowest of the waves called delta waves. And they range from 0.5 to 4 hertz. So last but not least is REM, rapid eye movement, or dreaming sleep. So interestingly, the pattern of EEG while you're sleeping is very similar to when one is awake. So I find that very interesting, very strange. So there's a lot of frequency information in EEG. So in general, the larger the brain volume contributing to the voltage of all of these of firing action potentials, the slower the frequency, and the smaller the brain volume that is contributing to oscillatory behavior is usually higher in frequency. Delta here is the slowest, and it's the strongest during deep sleep. So theta waves here are faster than delta, and it's, like I mentioned, it's strongest during drowsiness. The alpha wave is the strongest during relaxation, and it also shows up in things like inhibition control. So example, if you're trying to do a free throw in basketball, your brain is trying to ignore distractions and allow you to focus on just one task. And so the alpha signal shows up strongly when you're trying to ignore other inputs of sensory input. And beta, which is at the top, has the highest frequency and usually not a strong amplitude. And this beta is appears when one is actively thinking, and when one and obviously it's not a sleep signal, it's when one is awake. Okay, so I've given you a little bit of feeling of, well, where do these EEG signals come from, and what do they mean? So now we're going to talk about, well, what are the methods that we can use to analyze these oscillators? So here's an example of time domain analysis of an event-related potential. So this type of neuroscience research goes back decades to the 50s. An event-related potential measures the brain's response to an external stimuli, usually an auditory tone or visual stimulus. Each time a stimulus is presented to the brain's response is recorded via EEG, time-locked to the stimulus. So we can see here that at zero time is usually the point in time when the stimulus is presented. And this signal here is an average of dozens to hundreds of EEG signals, time series that are all averaged like 1 point at a time. So over the years, different associations have been found with the different peaks and valleys of the event-related potential. A famous one is called P300, and it occurs when the brain is surprised. So one way of inducing surprise in the brain is to create a series of tones, a common tone and a rare oddball tone. So I'm going to try to do that for you. So beep will be like the common tone, and a bop will be a rare tone. So a person's wearing an EEG headset and they're listening to tones that sound something like this. Beep, 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 bop, beep. So the P300 will show up for the oddball tone, which is the bop in the example I just gave, which is the surprising tone because it happens rarely. So an interesting thing about is that the P300 can be used as a measure of brain health. So it's going to occur even if one is in a coma, as long as their brain is still paying attention and is healthy. And this can show if like someone in a coma is in a locked in, with so-called locked in syndrome. They're awake, but they're in a coma. So this type of analysis is computationally simple. And so all we're doing is averaging single voltage values. And there's a wealth of literature that's been produced. However, there are a number of disadvantages with this method. It has a very poor signal to noise ratio, and hence the need for many time series to average. It's also very difficult to tie these responses with the underlying biological mechanism in the brain. And lastly, if there's a phase difference between the trials, then the averaging will flatten the peaks and valleys of the response. So next I want to talk about frequency domain analysis. So here's an example, and it's calculated using fast Fourier transform. However, convolution with wavelets can also be used and is commonly used. So this is still a very popular method of analysis because there's a lot of literature that associates the frequency and amplitude of oscillations with different brain activity. In this diagram, we can see an oscillation at the alpha peak here. The frequency and power of the alpha peak differs across people. The function of alpha oscillations also differs depending upon where in the brain the signal originates. So an easy experiment with alpha waves is to measure a person's visual processing center, which is located at the occipital lobe, which is located at the back of the head, about here. When a person's eyes are open, the alpha peak is very weak at the occipital. And this is because there's a lot of unsynchronized activity happening while the occipital processes complex information of the visual field, because the occipital is the center where our visual processing happens. When a person closes their eyes, there is no external stimuli arriving at the occipital. So when this region goes quiet, it starts to oscillate at the alpha frequency. Often the alpha waves are so strong that they can be seen by the naked eye just by looking at the raw data. And the alpha wave also is common for other sensory processing areas of the brain when those sensory processing areas of the brain go quiet. So when we see strong alpha oscillations in a brain region, it usually indicates that the brain region is quiet. So frequency domain analysis is very useful. However, a major drawback of frequency domain analysis is that temporal information gets lost when the FFT is computed. So that's why we prefer to use time and frequency domain. So this is an example of features that capture both time and frequency. This is called the spectrogram. And each color represents the power of the detected signal. So 1 pixel is the power of the voltage at a particular epoch in time at a specified frequency. So the y-axis is frequency and the x-axis is time. So higher numbers on the y-axis are faster oscillations, that is higher frequencies. And colors closer to red mean that the amplitude of the signal is stronger. So this spectrogram relates different stages of sleep with the activity of the hippocampus. During REM sleep, we can see here a strong theta oscillation, which lasts for the duration of this time period from here to here. So theta oscillation in the hippocampus is linked with forming or navigating memories. So time frequency domain analysis of EEG has many advantages. So it's closely related to the underlying neurophysiology of the brain. It allows for strict definitions of hypothesis testing, and the pretty colored plots and allow for exploratory analysis and visualization. Right? So say we wanted to create a classifier to predict different stages of sleep. How can we engineer features of the time frequency domain to feed into a classifier? So if I was in a lecture hall right now, I'd be looking into the audience to see if I'd see anybody raising their hands, but I'm not going to do that. But so I'll give you the answer. So this looks like an image to me. So why don't we use a state-of-the-art image processing technique? So the current state-of-the-art in image recognition uses convolutional neural networks. And here we see an example of classifying images of different vehicles, such as cars, trucks, bicycles, etc. There are two big stages in this example of deep learning classification. Stage one is feature learning, and it spans from here to here. And so stage one has two layers of convolutional neural networks, right? So although there's four matrices here, I'm going to combine, when I talk about a layer, I'm going to combine the convolution layer and the pooling layer into one. The second stage here is classification, which is done using fully connected layers of artificial neural networks. So starting at the left is the input. That's the bitmap of the image. And so there's one layer per color. So they'll, like, for instance, red, green, and blue. So this layer here is the first layer of convolution. So the height here and the width there represents the features that it's trying to discover. And the depth of it here is the number of filters. So a filter is shown here, and it's convolved across the layers of the color bitmap. In the diagram, it shows how one value in the convolution layer is calculated by convolving a filter with the image. So we can see that here by, here's the filter that's convolving with the image, and all of that convolution gets written into one value in this layer of layer of this matrix here in the convolution feature map. So more than one filter can be used in a layer, but the filters are all of the same size. In the next layer here, pooling is shown. So maximum pooling or max pooling is usually used, and this technique takes the largest value of a section of the convolution layer. And so this reduces the size of the map, of the feature map, into a smaller set of features. So here we see that example here where this is the max pooling, and all of those values, which are a part of a matrix, get written down into one value here in the pooling layer. And we can also see here that the size of the pooling layer is smaller than the number of values used in this feature map here. So in this example, a second layer of convolution and pooling takes place. So the two layers of convolution, they're learning features of the images. And they're learned using supervised learning by feeding the network labeled images of vehicles. In image recognition, the features discovered by the convolution tend to be things like vertical or horizontal edges or bright spots. The raw images get filtered by convolution and pooled, and to create a set of features that is smaller than the original image. So again, notice how pooling shrinks the size of the feature matrices. On the next layer in the convolutional neural network, again, the features get filtered and shrunken again by pooling. However, the features become more complex as we've used many more filters, as shown here by the width of this pooling layer. So the lower layers of convolution capture simple aspects of the image, such as edges or bright spots. And the higher layers of convolution capture more complex shapes and patterns, such as wheels or windows. So here in this example, there's only two layers of convolution that are shown, but here in the ... they're usually in complex image recognition. There's many more layers of convolution, and this rule of simple features to more complex features applies, and more complex features require a higher number of filters. And finally, here's this stage here in classification. It uses traditional fully connected deep learning networks to classify the features that are discovered by convolution into different vehicles here. So backpropagation with gradient descent is used to learn what the values of each filter to use to maximize the classification accuracy. So when training these networks, the coefficients and weights for the filters and fully connected layers they're first randomized. And then an image is fed into the network, and the error between the predicted value here and the actual value of the image is calculated. Then the weights and the filter coefficients are adjusted in proportion to the negative of the gradient of the error with respect to the weights. So if there's a large error, the weights will be adjusted strongly in a direction that will minimize the error. And this continues until the error converges to a minimum value. So this is computationally expensive and time consuming. So deep learning methods need a lot of computing power. And so an example is like, you need a stack of GPUs, and now there's a lot of cloud providers that are providing cloud solutions that will give you the computing power that's needed to train these networks. They also need a lot of data. So I gave you like a high-level introduction in the previous slide of what convolution is in image recognition. So in this slide, I want to zoom in on what's happening with convolution. So this 4x4 matrix here is a small section of a spectrogram. It shows two strong values highlighted in red, which indicates an oscillation of interest. So in this case, I'll convolve a 2x2 filter across each section of the spectrogram. So I've chosen this filter, these filter coefficients, 1, 1, 0, 0, to find horizontal edges. However, note that these coefficients will be learned by the training data using gradient descent backpropagation when implementing convolutional neural networks. So what I do, or what's done in convolution, is I place the filter at the starting point, which is the top left corner. Then I multiply and add up the results. So -1 times 1 plus -2 times 1 plus 0 times 0 plus 12 times 0 equals -3. So then I take the filter and slide it over 1, overlapping with the previous values. So here's the placement of the filter again. So then I repeat the same multiplication of each element here to here to here. And when I add all of those up, I get -4. So then I keep repeating that. So I put the filter here and calculate, well, what's the value? And then put the filter here, and then on until I fill this 3x3 matrix. So that's the convolution part. So another step is needed to show activations. And the convolution values are passed through what's called a rectified linear unit, which means, and so what a rectified linear unit does, if the values are below 0, then they get zeroed out. And if they're greater than 0, then we just keep the values, the positive values. So we can see here that all of the negative values get zeroed out, and all that's left is the positive values. So these are the activations. And then I haven't shown it here, but there's another step called pooling, which I mentioned before. And so pooling is simply, so let's say I have a 2x2 pooling filter. I take that pool, the 2x2, go across here And what's the maximum value? Oh, it's 26. So that 26 is what's carried forward into the pooling layer. And again, here and here and here. So 4 values of 26 are going to show up, but this is a small example. Okay. So I better speed up. So here I'm showing an example of convolutional neural networks to classify stages of sleep. So there are 6 stages of sleep as shown in the bottom right, from awake to drowsy to light to deep sleep and REM sleep. So classifying sleep stages can help diagnose sleeping disorders. And so in this case, long recordings of EEG data were broken down into non-overlapping segments of 30 seconds. The classification will be given to each segment. So EEG data across 7 electrodes across the scalp were used. And the input data are spectrograms for each electrode. So we can see the inputs here, that each spectrogram is one matrix of frequency and time. And then there's 7 spectrograms. So the first stage of convolution and pooling are shown here, and we can see that there's a reduction in the size from the input to the first layer of convolution, and then to pooling. So we repeat this. We have 2 stages of convolution here. Well, by 2 stages, I mean convolution 1, pool 1, convolution 2, and pooling 2. So the number of filters needed in EEG is less than image recognition because EEG spectrograms have lower complexity than images. And I took this example from a thesis, a dissertation by a McGill student. So at this point, all of the features were calculated, and the classification of the features into stages is done by 2 fully connected artificial neurons here. So there's 24 artificial neurons here, 24 artificial neurons here, and they're fully connected to the previous layer. And then this layer is fully connected to this layer, which means that for every input, every neuron here, it has 24 inputs from here. And then the final layer is softmax, which just normalizes the classification into 6 probabilities, which all add up to 1. And so the value with the highest probability is the sleep stage that's classified. So convolutional neural networks do well in the literature compared to other machine learning methods, especially for classifying sleep. Okay, so here's another paper that used deep learning to discover features. So here, it's done a good job. So in this method, deep learning has discovered what a sleep technician would find when visually inspecting biological signals of sleep. So here are features of EEG. And in this case, in addition to EEG, eye movement is captured. So this is eye movement of the left eye movement of the right eye, and here's muscle tension. Okay, another feature of EEG that's often used is spatial information. So one technique to get spatial information is called common spatial pattern. And it takes 2 different states and compares their relative power across different electrodes. In this example, there were 4 electrodes. So this gives us a spatial pattern shown by the contours given in this diagram. And this diagram shows the spatial pattern for a wink compared to both eyes resting. So we can see here that the wink in the right eye has a lot of power compared to the rest of the scalp. So EEG also picks up sources of unwanted biological noise. So here's an example of raw EEG contaminated by muscle noise and a heartbeat. So independent component analysis is a method used in EEG to remove unwanted artifacts. So it's a method of blind source separation that works to recover the original sources that have been linearly mixed. So here we can see that the raw EEG data has been transformed into ICA components. And it did a good job of separating the heartbeat and the muscle tension, but it's not perfect. In this fifth component here, there's still evidence of heartbeat. And the idea was to separate out the unwanted biological artifacts just to get pure brain signal. Okay, so that was it for the evolution. I'm going to talk a little bit about Neurovine now. So digital health is transforming the healthcare industry to put the patient more in control of their own health. So people have devices now that measure their heart rate and their blood pressure, et cetera, that they use to help them manage their own health. However, there's no devices available for helping patients manage their own brain health. So the vision of Neurovine is we want to put brain data into the hands of individuals so that they can take more control over their health. So we're using evidence-based medicine to create solutions for the brain health market. So our first product is focusing on helping people recover from concussion. At Neurovine, we're doing a lot of interesting digital signal processing and machine learning to help people recover from concussion. So I'm asking you to consider joining our research study. We need to measure the EEG of healthy brains so that they can be compared to concussed brains. So if you join the study, we'll put your brain through a number of different exercises while recording your EEG. So if you join the study, you can learn more about neuroscience and EEG. So this is work from Neurovine as well. So the status quo for recovering from a concussion is to sit in a quiet, dark room and rest. So this doesn't work well. We now know that moderate mental effort will help patients recover more quickly, but only to a point. If they push themselves too hard, then a patient's recovery could regress. So at Neurovine, we're developing a product that allows concussed patients to know when they need to rest their brain. And this is the brain energy graph. It shows how brain energy depletes as the user increases their mental workload. A metaphor for brain energy is like the amount of energy in a battery. As current is drawn from the battery, reduces its total energy is like when one uses mental effort in thinking. So as brain energy depletes, it goes from green, meaning high brain energy, to yellow, meaning moderate, to red, meaning stop and take a rest. The y-axis shows brain energy, and the dashed red line is the threshold of brain energy that will create a brain break alert. The x-axis is time in minutes. So the brain break alert happens here, where brain energy crosses the threshold joined by the dashed red line. The tasks that the user is doing are shown in the text along here. We can see here that the user was doing a task that required a lot of mental effort because how rapidly the brain energy declines at this point. They were putting a lot of effort into thinking. It also happened here and here. So we can also see here an example where the user was resting and the brain energy stays flat, meaning that it didn't deplete, their brain energy didn't deplete very quickly. The orange line here shows their self-report of fatigue or how tired they were. And the higher the number here, the more tired the user was feeling. We can see that this user's brain break alert happened well before there was a long period of fatigue. So this is an example of what we're doing at Neurovine to help people recover from concussion more quickly. And the idea is that they use this and they'll do their work or tasks, sit at the computer, whatever they need to do, And they can do that with confidence, knowing that if they're going to push their brain into an area that they're going to harm themselves, then a brain break alert is going to warn them to stop and take a rest. So we're looking for healthy brains to be part of our study. So I'm asking each of you to consider joining our study. And the study has been approved for the community, as well as we can recruit within Carleton University, as shown here. Before I move to questions, are there about the talk? Are there any questions about the study? Okay. Okay, thanks. So I'll take questions now about the talk about Neurovine, but I'll leave this slide up so that you can record the contact information shown here.
Thank you, Lou. So,
Hey.
Lou, so now we
Welcome.
will open the floor for discussions and also questions. So the choice is either you can write the questions, or if you want to ask the questions yourself, could you please raise your hand? And I think in the participants, if you go and then raise your hands, it will show up, and then I can call you. Okay. All right, so let me start with a question, because I haven't seen anybody post a question yet. So Lou, in most of the cases, as you have said, people treat the STFTs as images. So I have a question here. Image is different from an STFT because images go from, say, 0 to 256 in the case of a gray image, for instance. Whereas STFT is not like that. They are all values. And so to treat an STFT as an image and then do the processing, we are losing a lot of information here in the process. So why should one use it as an image and not rather use it as a spectrogram or an STFT and send it through the CNN? Because there is no reason why a CNN cannot take an STFT.
Yeah, you're right. And so the reason why I chose to provide a spectrogram in my talk was because it just flowed well in terms of how I was presenting the evolution. So there's no reason why you couldn't apply, and in fact, it probably will work better if you apply a convolutional neural network directly to the raw voltage values of the EEG itself, and it can be trained, and the filtering coefficients will be different for real-time series of voltage values, but that's a good way of doing it as well. And actually, if you wanted to give the frequency components, which also include like the phase information in the full short-term frequency, that is also can be done as well. So I guess you could treat like the imaginary and the real part of a Fourier transform as like two layers of it, and then apply a convolutional filter to that. So that's a valid way of doing it as well. And I just chose one way here just so that it was an evolution in terms of how I presented the material.
So before I go into the chat with other questions, there's one more thing that I want to pick your brain on. In most of the papers, people start with using AlexNets or other traditionally used nets. And the input seems to be fixed there to certain weird numbers like 223 on columns or rows, and then let's say 170 on the rows or something like that. And naively, people take whatever the STFT that they have, for instance, and then make sure that they fit to those numbers. And in my opinion, I think it's a no because a lot of things are happening here because we may be erasing things and then moving forward. So what is your opinion on that, and what are your suggestions to the students here, whether they should blindly go and then take the architecture of AlexNet and then fit for instance, a square peg in a circular hole and then try to do their work? Or what's your viewpoint on this?
Well, I guess they can use an existing architecture just to see how it works, because everything, well, I guess everything is pre-trained, and so they can in many of these, everything is pre-trained, or you can retrain the weights using the existing architecture, and that's fine, because that's a good starting point. But I guess it depends upon the experiment. And so the data scientist that is using convolutional neural networks and deep learning has to use their judgment and experiments to guide them in terms of, well, what's the right architecture? And this takes iteration, right? And also another thing that I would recommend that is done is there's ways of visualizing what features are being discovered. And so you should, at each layer of convolution, you go, well, what is it doing? Like, so used to be that artificial neural networks weren't transparent. So now there's techniques to see, well, what's happening at each layer? And so look at what are the features that each layer is finding, and does it make sense for the problem that you're trying to solve?
Thank you, Lou. At least we will stop people doing monkey see, monkey do business here. Thank you. So there is one question from Emma Farabo, and she's asking this. Can you tell us more about how the Neurovine headset works? How many electrodes does it have? And how does it compare to using the full cap?
Okay, so Neurovine has the Neurovine headband has 4 channels, and it fits around your head, and it attaches. So the electrodes attach to the ears, as well as there's electrodes on the forehead. But all of those, it gives 4 channels of EEG data, and so we're getting very high signal quality. We're using state-of-the-art electronics, and with good resolution and with low noise and good accuracy. And they're dry. They're we're asking people to put saline water on the electrodes when they attach it to the forehead, and that gives really good low impedance, and the person can wear it for like about 2 hours, and good signal quality is given. With a full cap, you need a technician. You just can't put a full cap on yourself. So a technician is needed, because each electrode has to be adjusted individually, and usually conductive gel, extra conductive gel, it needs to be squirted into each electrode to get through hair to make contact, good electrical contact with the skin. So depending upon the number of electrodes, it can take up to half an hour or more for a technician to put the cap on your head. Whereas Neurovine is designing a device that you can use on your own.
Thank you, Luo. Mohammed Abdul Aziz has a question, and I believe he wants to ask it himself.
Yeah, if that's okay, thank you.
Sure.
First of all, thank you for the presentation. I had a question regarding the type of networks that could be used. So you've discussed CNN as an approach that could be used. However, I was wondering if you have considered any architectures that would take into account the time information in EEG. And by time here, I don't mean like using STFT, which includes time, but I mean previous epochs as an example. Because I don't believe someone who is awake, as an example, could go directly into a REM sleep stage. They have to first transverse through the other sleep stages. So can you comment about if you have seen these type of networks out there, and or do you think they're beneficial, or just using an epoch by itself would be good enough?
Yeah, that's a good question, and that's a good insight that can be added. And so, especially in sleep, because there's a transition from one state to another. You don't go from being awake 1 second to being in deep sleep. You transition through stages. And so using information of the previous state can help you predict what the next state is going to be. And yeah, there are techniques. I didn't mention them in my talk. One of them is hidden Markov models. And I've seen a paper that uses that and in combination with deep learning to classify sleep stages. Another network, which is a deep learning method, is recurrent neural networks, and it uses prior information to help classify the current time. Yeah, so yeah, the answer to your question is yes, you can go back and look back into time and see what happened before the current time period to help you make a classification. That's valid for sure.
Perfect. Thank you for the information.
Thank you, Lou. There is another question from Dr. Hamid Reza Sadrizami. So his question goes like this: EEG signals have very high variability, the intersubject, and generalization is challenging. It is almost impossible to apply CNN directly on the time domain, or even STFT is not very informative in this sense. Can you please elaborate more on how you have handled this variability and the very low SNR of EEG signals?
Yeah, so to handle the low signal to noise ratio, the EEG signal needs to be clean. And has sources of biological noise and environmental noise, movement, such things, bad epochs, have to be removed. And so at Neurovine, we've created a classifier that identifies when the EEG is clean. That is, when we know for sure that this epoch is purely brain signal. The other part of your question is variability. How do we deal with variability? And between individuals, yes, everyone's brain sort of, there's commonality, just like there's commonality in our DNA, but there's also a lot of variance in our DNA. So everybody's brain is different, and so what we're doing at Neurovine is we're customizing our models per individual. So if you join our study, we're going to train a specific model for your how your brain responds to the exercises that we would give you.
Thank you. I don't see any other questions on the chat. If anybody has any burning question, kindly ask on the chat. If not, we will be okay. Someone says that they joined late, so missed few minutes of the discussion. Oh, that's for me. All right. Okay. They were asking about the recording later on to refer. Mrinmai, are you a Carleton or University of Ottawa student? Okay. So if you're a University of Ottawa or Carleton student and you're part of this biomedical engineering seminar series, you have access to this recording. And if you're not, then probably you don't have an access to that, because this has been recorded for the private use. And unless Dr. Pino allows me to make it available to everybody, it won't be made available. Thank you.
Yeah, we're gonna make it public anyway, so that's fine.
Okay, so when Dr. Pino's company makes it public, it will be available to you guys. Okay. Okay, there is one more question. I have a question regarding the application of the research. Do you have to have a resume ready? I think they are getting a different information, I believe. So this is about, let me allow Dr. Pino to address this.
Okay, yeah. So we're looking, we're doing like a clinical, not a clinical study in this case, but we need information to EEG information to help us develop our algorithms. And so we went through Research Ethics Board, created a study. And so what we're looking for is people to participate in a study. And so you sign up, you sign, we tell you what the study is about, and then you sign up for it, and then researchers will come and virtually in this age of COVID, we'll send you a headband, and then you put the headband on. We'll tell you how to put the headband on, and then they'll guide you through some exercises that you do over your computer. And while you're doing these exercises, we're recording your EEG. So if you're looking for information, more information, you can look in the website here, or email this email here, research@neurovine.ai, or there's a number of other social media ways to get in touch with us. But yeah, so we don't need your information in terms of your resume, but I think they'll need to know, are you over 18? Have you had, we want a healthy brain, so we just want to ask that, is your brain healthy? Have you, no recent concussions that have happened, those sorts of questions. And then if you agree to do the study, we'll get signed up and we'll schedule a time to record your brain.
Yeah, just a clarification. So you'll be let me quote unquote say that you'll be donating your signals to Neurovine. So you are a participant. So this is not a job posting. So hope that clarifies.
Yeah, that's correct. Yes.
Okay, excellent. Any other question? If not, let us give a big hand to Dr. Pino for this wonderful talk. And let's also wish him good luck, and the company also good luck in their research. And it's a startup in Ottawa, and they have started during their studies now to get the product going. So hopefully they will have a product soon, which then we can borrow for doing our research. Thank you. Thank you, Pino.
Okay, you're very welcome. Thank you for having me, and I hope you and your students benefited from my talk.
Thank you so much.
Okay. You're welcome.